Hello, and welcome to the summary of Chapter 15, Genes and How They Work. The genetic code is read in groups of 3 nucleotides called codons. There are 64 possible codons. 3 of them, here, here, and here, represent stop, while 1 down here represents start. Now, since there are only 20 amino acids and we have 64 possibilities for codons, there are many codons that code for the same amino acid. For example, serine has 4 possible codons, and you'll notice they all start with the letters UC, or the nucleotides UC, and the third nucleotide is different. The code is the same in almost all organisms, which supports a common evolutionary heritage, and genes can be transferred from one organism to another and still function in the new host. In the case of these pigs, the piglet on the left has a jellyfish gene that encodes a green fluorescent protein. When a gene's genetic code is read and used to make proteins, and then those proteins then carry out their function, we say the gene has been expressed. Gene expression involves several steps. The first is transcription. Transcription is the process of making a copy of the genetic code on the gene. This is done using an enzyme called RNA polymerase. There are different forms of this enzyme depending on whether it's a prokaryotic or a eukaryotic cell and what type of gene is being copied. RNA polymerase will bind to a special site on the gene known as the promoter. It will then be able to determine the start site of where to begin copying the gene. The region that is copied is called the transcription unit, and at the end of the transcription unit there is a terminator sequence which tells the RNA polymerase where to end the copying. Having a promoter and a start site are important not only for determining where to begin transcription, but also what direction to go in. The code is copied by building a strand of messenger RNA. In the figure here you can see the new strand being built. It grows from the 5' to 3' direction. During this process, the RNA polymerase actually unwinds the DNA strand and unzips it. So this whole structure, the RNA polymerase, the template strand, and the messenger RNA strand that's being built, is referred to as the transcription bubble. DNA is rewound as it leaves the bubble. There are some very important differences in the mRNA strand between prokaryotic and eukaryotic cells. For example, in prokaryotic cells, the mRNA strand may code for several related genes and not just one. In eukaryotic cells, it is the enzyme RNA polymerase II that makes the messenger RNA strand, and this is known as the primary transcript. The primary transcript must be modified in the nucleus before it can be sent out to be translated into a protein. A cap is added to the 5' end to prevent degradation and to assist in the initiation of the translation process. A series of adenines are added at the 3' end known as a poly A tail. This is also done to prevent degradation. Many eukaryotic genes contain non-coding sequences that have to be removed from the primary transcript to form the final messenger RNA sequence. The non-coding sequences are called introns, while the coding sequences are called exons. Small proteins known as SNRPs will cluster together and form a larger complex called a spliceosome. Removal of introns is known as splicing. A primary transcript can be spliced into different mRNAs by the inclusion of different sets of exons. This process is called alternative splicing. Alternate splicing explains how 25,000 genes in the human genome can encode for more than 80,000 different mRNAs. Once the mature mRNA is formed, it then leaves the nucleus and travels to the cytoplasm to be translated into a protein. When the mature RNA sequence reaches the cytoplasm, it will bind with a ribosome. The ribosome attaches to the mRNA at the 5' cap. The larger subunit of the ribosome has 3 sites where tRNA can bind. They are known as the E, P, and A sites. On the tRNA, you have a 3 nucleotide sequence known as the anticodon. The first tRNA anticodon will bind in the ribosome to the mRNA codon at the P site. The next tRNA binds to the A site. The tRNA in the P site will transfer its amino acid to the tRNA in the A site. The ribosome will then shift down the messenger RNA strand, moving the tRNAs to the E and P sites. This allows for a new tRNA to bind to the A site. This process continues, building the amino acid chain until the stop codon is reached. There is a short video posted in Blackboard under the Unit 4 folder which illustrates how this process works. The end of Chapter 15 describes the different types of mutations that are seen in the DNA. Under the category of point mutations, you can have base substitutions which affect a single site in the DNA. This includes a silent mutation, meaning the codon still encodes for the same amino acid, or it could be a missense mutation where the codon is changed to encode a different amino acid, such as in sickle cell anemia. Nonsense mutations change the codon to a stop codon, and frame shift mutation is the addition or deletion that shifts the reading frame. In triple repeat expansion, there is a triplet sequence normally found in DNA that gets expanded. This can be seen in Huntington's disease. Mutations can also occur in large sections of chromosomes. You can find deletions, as in the condition known as Cri-du-Chat, where there's a missing part of chromosome number 5, or duplications, repeated segments of chromosomes. Translocations are exchanges between non-homologous chromosomes, as seen in 2 forms of leukemia, and inversions are when a segment of a chromosome is turned 180 degrees. While it seems as though mutations are a bad thing, they are actually a necessary tool used to help increase survivability of a species. It is the mutations that give rise to new adaptations, allowing individuals to be better suited to surviving in their changing environment. This is the underlying process of evolution.